The theory of spontaneous generation. In the 1600s, two scientists on opposite sides of the theory of spontaneous generation, one believer and one non-believer, battled it out in the lab. Francesco Redi was a non-believer. However, the majority of scientists, as well as lay people, believed in spontaneous generation. Antonie van Leeuwenhoek contributed greatly to the history of microbiology in a lot of other aspects. He was actually a great proponent for spontaneous generation. So what Redi did is he performed a simple experiment using jars containing rotting meat and covered one of the jars with gauze to prevent flies from coming into direct contact with the meat, while the other jar was just left open. So one of the ideas of spontaneous generation was this pneuma, or vital force, which was believed to be that spiritual force that led to the spontaneous generation. The pneuma was believed to travel through the air. This is why gauze was used to close one jar and not the lid. So let's look at the possible outcomes. If spontaneous generation was correct, both samples of meat should produce maggots because they are both exposed to the air. So the pneuma, or vital heat, should be able to cause spontaneous generation. If spontaneous generation was incorrect, the meat inside the covered jar will not produce maggots. In other words, the presence of these microorganisms has nothing to do with pneuma or vital heat. Possible outcome 3 would be if maggots appear in meat only after there has been direct contact with flies. Then, the meat in the uncovered jar will produce maggots, and the meat in the covered jar will not produce maggots. The conclusions of Redi's experiments was that spontaneous generation is not correct. Another conclusion from Redi's experiments was that the maggots appeared in meat after coming into direct contact with flies, giving evidence to the hypothesis that maggots may come from flies. So this was the first evidence against spontaneous generation. Thank you for watching.